Introduction. Biochemical regulation is a fundamental process that ensures the proper functioning and coordination of cellular activities. It involves a complex network of molecular interactions that control gene expression, cellular signaling, and metabolic pathways. Understanding the mechanisms of biochemical regulation is crucial for deciphering various biological processes, identifying molecular targets for therapeutic interventions, and exploring the pathogenesis of genetic disorders. In this script, we will delve into the intricate details of different regulatory mechanisms and their significance in biological systems. Each topic will be explored in depth, providing a comprehensive overview of the regulatory processes at play. Let's begin our exploration with the process of transcriptional regulation. Transcriptional regulation plays a vital role in controlling gene expression. It involves the process of converting DNA into RNA by RNA polymerase enzymes. However, not all genes are expressed at the same time or in all the cells of an organism. This is where transcriptional regulation steps in. Various factors and mechanisms work together to determine which genes will be transcribed and to what extent. Transcription factors, such as activators and repressors, bind to specific DNA sequences known as enhancers and silencers, respectively. These transcription factors either enhance or inhibit the binding of RNA polymerase to the promoter region, thus modulating transcription. Additionally, chromatin structure and epigenetic modifications play a crucial role in transcriptional regulation. DNA methylation and histone modifications can either activate or repress gene expression by altering the accessibility of DNA to transcriptional machinery. The interplay between transcription factors, chromatin remodeling complexes, and epigenetic modifications ensures precise and dynamic control of gene expression. Even after transcription, gene expression can be further regulated at the post-transcriptional level. RNA processing, mRNA stability, alternative splicing, and translational control are some of the mechanisms that contribute to post-transcriptional regulation. RNA processing involves the addition of a 5' and a poly tail to the mRNA molecule, which influences its stability and translation efficiency. Alternative splicing allows the production of multiple protein isoforms from a single gene by selectively including or excluding exons during mRNA processing. Post-transcriptional regulation also encompasses microRNAs, miRNAs, and other non-coding RNAs that can bind to the mRNA molecule, resulting in degradation or translational repression. Furthermore, RNA-binding proteins play a crucial role in determining mRNA stability and localization within the cell. Together, these post-transcriptional regulatory mechanisms fine-tune gene expression and contribute to cellular diversity and response to environmental stimuli. Feedback inhibition is a common regulatory mechanism that maintains homeostasis and prevents excessive accumulation of metabolic intermediates. It involves the inhibition of an enzyme within a metabolic pathway by its end product. When the concentration of the end product exceeds a certain threshold, it binds to a regulatory site on the enzyme, altering its conformation and inhibiting its catalytic activity. This negative feedback loop helps maintain the optimal balance of metabolites within the cell. Feedback inhibition is an efficient mechanism to regulate metabolic pathways, ensuring that resources are not wasted and energy is conserved. In complex biological systems, multiple genes and pathways are often regulated in a coordinated manner to ensure appropriate cellular responses. Coordinated regulation involves the integration of different regulatory mechanisms to achieve a finely tuned response. For example, during stress conditions, various stress response genes need to be activated simultaneously. This coordination can be achieved through the interaction of transcription factors, signaling pathways, and chromatin remodeling complexes. Coordinated regulation ensures that all the necessary components for a specific biological process are activated in a synchronized manner, allowing for efficient cellular adaptation and survival. Hormonal regulation plays a critical role in maintaining homeostasis and orchestrating physiological processes. Hormones, such as insulin, glucagon, and estrogen, act as chemical messengers that bind to specific receptors on target cells to initiate signaling cascades that regulate gene expression and cellular activities. Hormones can exert their effects at the transcriptional level by modulating the activity of transcription factors or altering chromatin structure. They can also regulate post-transcriptional processes, such as mRNA stability and translation. Hormonal regulation is tightly controlled and ensures the proper functioning of various organ systems, reproductive processes, metabolism, and growth. Nutrient sensing mechanisms allow cells to monitor and respond to changes in nutrient availability. These mechanisms are critical for maintaining energy homeostasis and adapting to fluctuating nutrient levels. Nutrient sensing involves different signaling pathways and regulatory networks that integrate information about nutrient availability and metabolic status. For example, the AMP-activated protein kinase, AMPK, pathway senses the cellular energy status and regulates gene expression, metabolism, and cell growth accordingly. Another well-known nutrient-sensing pathway is the target of rapamycin, TOR, pathway, which integrates signals from nutrients, growth factors, and energy status to coordinate cell growth and proliferation. Nutrient-sensing mechanisms are essential for proper nutrient utilization, energy balance, and overall cellular health. While genetic information is inherently stable, epigenetic modifications provide an additional layer of regulation that can be modulated by various environmental factors. Epigenetic regulation involves heritable changes in gene expression that occur without alterations in the underlying DNA sequence. DNA methylation, histone modifications, and chromatin remodeling are some of the epigenetic mechanisms that regulate gene expression. These modifications can be influenced by environmental factors, such as diet, stress, and toxins, and can have profound effects on development, aging, and disease susceptibility. Understanding epigenetic regulation is crucial for unraveling the mechanisms underlying complex diseases and developing targeted therapeutic interventions. Enzymes are central to almost all biochemical reactions and are usually subject to regulation to maintain metabolic homeostasis. Allosteric regulation is one of the mechanisms by which enzymes are modulated. It involves the binding of small molecules, known as allosteric effectors, to specific sites on the enzyme, away from the active site. Allosteric effectors can either enhance or inhibit the enzyme's activity by inducing conformational changes that affect the enzyme's catalytic efficiency. Allosteric regulation allows for rapid and reversible control of enzyme activity in response to metabolic changes or signaling events. This mechanism enables cells to fine-tune metabolic pathways and respond to varying physiological demands. Cofactors are non-protein molecules that assist enzymes in catalyzing reactions. The availability and regulation of cofactors play a crucial role in modulating enzymatic activity. For example, vitamins and minerals are essential cofactors that are required for proper enzyme function. The deficiency or excess of specific cofactors can lead to enzymatic dysfunction and disrupt metabolic pathways. In some cases, regulatory molecules can act as cofactors, influencing enzyme activity by binding directly to the enzyme or affecting its conformation. Cofactor regulation ensures that enzymes have the required components for their optimal catalytic function, contributing to metabolic control and overall cellular health. Cellular signaling pathways enable cells to communicate, respond to stimuli, and coordinate their activities. These pathways involve the transmission of signals from cell surface receptors to intracellular signaling molecules, eventually leading to changes in gene expression, metabolism, or cellular behavior. Signaling pathways can be activated by a variety of extracellular ligands, such as hormones, growth factors, and neurotransmitters. They often involve cascade-like amplification mechanisms, where a ligand-receptor interaction triggers a series of molecular events that amplify and transmit the signal within the cell. Examples of well-studied signaling pathways include the mitogen-activated protein kinase, MAPK, pathway, the phosphoinositide 3 kinase, PIEK, pathway, and the cyclic adenosine monophosphate, CAMP, pathway. Cellular signaling pathways are essential for cellular homeostasis, development, immune responses, and cell survival. Genetic disorders result from alterations in the structure or function of one or more genes. These disorders can be caused by various mechanisms, including mutations, chromosomal abnormalities, and epigenetic modifications. Understanding the underlying genetic basis of these disorders is crucial for diagnosis, prognosis, and the development of targeted therapies. Many genetic disorders involve dysregulation of biochemical pathways, leading to metabolic imbalances or defects in vital cellular processes. Research in this field aims to identify the molecular mechanisms underlying genetic disorders and develop innovative approaches to prevent, manage, or cure these conditions. The knowledge gained in biochemistry and regulatory mechanisms has paved the way for numerous medical applications. Understanding how regulatory processes work and how they can go awry in diseases has led to the development of targeted therapies and diagnostic tools. Pharmaceutical companies utilize this knowledge to design drugs that can specifically target dysregulated proteins, pathways, or genetic alterations. Additionally, biochemistry and regulatory mechanisms play a crucial role in personalized medicine, where treatment decisions are based on an individual's genetic makeup, lifestyle, and disease characteristics. Furthermore, the field of genetic engineering and gene therapy relies heavily on our understanding of biochemical regulation to develop gene editing tools and deliver therapeutic genes to treat genetic disorders. The applications of biochemistry and regulatory mechanisms in medicine are vast and continue to advance with ongoing research and technological developments. Biochemical regulation is a complex and fascinating field that governs the delicate balance of biological systems. transcriptional and post-transcriptional regulation, feedback inhibition, coordinated regulation, hormonal regulation, nutrient sensing, epigenetic regulation, enzyme allosteric regulation, cofactor regulation, cellular signaling pathways, genetic disorders, and medical applications all contribute to the intricate web of regulatory mechanisms. Understanding these processes is crucial for deciphering the intricacies of life, developing therapies for diseases, and exploring new frontiers in medicine. Continued research and exploration of biochemical regulation hold immense potential for unraveling the mysteries of life and improving human health.